All right, there we go. All right, just in case. So here's what you need to do for this week. So here is your analysis and interpretation. Before you answer these questions, this video that I linked with you is just a short video on these 3 words. If you rather watch a video to learn words, then read my background information. Because these words, hypertonic, hypotonic, and isotonic, you need to know what those are, because every single cup you have to answer, was it hyper, hypo, or isotonic? Okay, so here's a short video to watch for you guys on that. Okay, your data table is worth 25 points. So just to show you, out of the 100 points, this data table is worth 25 filled in. So make sure you guys have this all done and all this filled in for me. It's worth 25 points. Okay, if you look at this, each one is worth 5 points. 5 points, 10 points, 15, 20, 25 points. Okay, so that's 25 points of your lab as a data table. Now here's where you have to do a little bit of modeling, because this is modeling diffusion and osmosis. Okay, so first you need to go into your folder and make 5 copies of this for you right here. Okay, so you need to make 5 copies of this. How do you make a copy? Right click it, okay, and you go down here and make a copy. All right, so if I make a copy of that, so see my copy here. Obviously you have to make your own and don't put it in this folder. But here's what you have to do next. You have to label your copy, cup number 1, your first one. So you go back, and now I have a copy of it. And again, don't put it in this folder, because I don't want to see tons and tons and tons of copies in here. This needs to be into your own folder. And you need to rename this one. So right click it. So again, I right clicked it. Go down here to rename, and I rename it cup number 1. Okay, you're gonna make 5 of these: cup number 1, cup number 2, cup number 3, cup number 4, and cup number 5. Okay, I'm just getting rid of that because this is just an example. So you've got 1, 2, 3, 4, 5 labeled. You got 5 of those. Now, here's what you need to do with this. You need to move the pieces or manipulate them to what happened with your experiment. So let me show you that, okay? So in the video, you are going to explain to you hyper, hypo, and isotonic, okay? And so these pieces can be shaped, you're using Google, and resized to what you need, okay? You're explaining what happened to me through a model, so I can see it right away. So let's go back to your first one really quick. Oops, you're supposed to do. Your first one was supposed to be salt water in cup number 1 and water on the outside. So here's how you do this. Now everyone, pay attention. So if you're doing something else, focus, because I want you to see this and what I'm doing, because you need to see this, okay, to model this, how to use this. So I've got salt inside and water on the outside. Let's go back to my model, okay? And I've got salt on the inside, okay? So here's my cell on the outside, and I've got salt water on the inside. So what I can do is I can rename this salt. See that? And I can put it inside a cell. And I can make more salt, because you're gonna have to tell me if it's, you can make rename all these salt if you need to, and move as many as you want. Whoops. Okay. I'm just renaming them and clicking on renaming them. Okay. And I can like highlight all of these and move them down if I want. It's just things to use. Okay. This is a cell. What is outside of the cell? Anyone know? Outside of our cell number 1 was what?
Water.
Water, right? So this right here, you can change the color if you want to make it blue, but you can right click it, okay? Or if you just clicked on the whole thing, you can go over here and see how this yellow is like right here on the yellow fill color. You can change that to blue or something like a light blue if you want, okay? Now, the cell this is like your bag, right? Inside of the bag. The cell is into the water. So I need the water to actually be behind it, don't I? See how the water's like going in front of it? So this is what you have to pay attention to right now. If I right-click this whole thing, I go down to this word called order. And order means like, is it in the front of the picture or in the back of the picture? So since this is the surrounding environment, I want this to be in the back. So like the cell can kind of like be inside of it. So I send this to the back and watch what happens. If I move it over, it's magically behind it. See that? Okay. And now I can just make it bigger. So I can do that. And now the cell is kind of like inside of the water, right? So the surrounding environment, and you can just change this if you wanted to be water. See that? So this is now water, and this is the inside of the cell. Okay? And you can just move this around. It's just a text box. All right. So now, what do you need to do? And this depends on what happens. Since you've got salt inside of here, okay, starting out, and this is your cell, and you've got water out here. You could even put water, these are like water molecules. You could even put them over there if you want. Okay, you got water molecules here. What's happening? Okay, because you don't know until you do this, did salt go out? Okay, so you can so I just move that arrow. Maybe you thought salt went out of your cell. So then you would move this, and on the arrow or above the arrow, and you can make this smaller, bigger. But did salt go out of the cell? Maybe that's what you thought would happen. Or maybe water went into the cell. So then you could do something like this, into the cell, and you could just move like, water is going into the cell or something. Water is going into the cell if you wanted, okay? So you need to obviously do the experiment first, okay? So I want to stop you here because there's one more thing after this, but who has a question on just moving these around? If you want me to show you something again, who has a question on how to move this stuff? Because that's what I want to make sure you guys understand, because you're going to be doing this before Christmas break. It should be done. I won't see you again until next week. So I want you guys to do this. Anybody? Want me to show you something again that you didn't understand how to do it?
So basically, after we do the experiment, this is just making the model of it to represent what we did.
Yep. But you're going to do 5 of these.
5?
Yep.
Why 5?
Because here's why you're doing 5 of these.
Oh, right.
5 cups. What happened with your salt cup and water cup? So again, remember, this one will be labeled cup number 1, right? I would have to relabel this one cup number 1 because this is representing your first cup, okay? And I don't know, obviously I know what should happen, but you don't know if it's gonna go in or if it's gonna go out, okay? You don't know yet, okay? So let's say this is what's happening. Water's going into the cell. So what you need to do then is show me that. If water's going in, oops, you can't see it, I'm sorry. I stopped sharing. You can still see it? Oh, okay. I thought I stopped sharing. So if water goes into the cell, sorry, you need to get this double arrow right here, and then you can move it into there, okay? So if water's going into the cell, I need to put a bunch of water molecules into the cell for my model, okay? It's going into it. So all the water is then gonna go into it. Okay, so now look, I've got only like 2 salts, which obviously there's like thousands of salt molecules and thousands of water molecules. But let's just say this is just a model. You're supposed to show that there's more water than salt. Does anyone know if there's more water than salt, what do I get? Is it isotonic, hypo, or hyper? Let's go back here. So this is what you do. If you don't know, you need to go back to your background information. Let's figure this one out. All right, hypo, higher concentration of water and a lower solute concentration. Hyper is a low concentration of water and a high one of solute. So here's a little table. Water concentration high, solute concentration low for hypo. So this one has more water inside the cell. So what is it?
Hypo.
Hypo, yep. So then you would, on the cell, you go over here and put the word hypotonic, because this cell is hypotonic. There's more water in there than there is salt. Okay? All right. So this is what you would then turn into me. Okay, you can turn this whole thing into me, but because you just got some leftover stuff here. But I'm looking for something like this. Obviously, all these are moved away. Okay. But this is what I'd be looking for your first model then, if this was true. Okay. Water is going into the cell, or you could even move the water over here if you want to be more specific. The water from here is moving into the cell. Okay. And so you could even do it like this. You could just have the arrow go, boop, water's going into there, and it's going into the cell, okay, with the arrow. So it's just saying water's going into it. And then it's more water. You have to show me that there's more water than there's salt. You have to tell me what's here, hypotonic, and then you have to tell me what's on the outside. So this is what should happen with one of your models if this is true. Okay? All right, last thing. I'm done talking. Too much talking by Mr. Jackson. Then, once you're done with that, obviously you have to turn those into me. You could screenshot it. You could actually turn the physical ones into me on Seesaw. But then you've got to interpret it. So the last thing you have to do is answer these questions for each cup, which is super simple. Did your model cell gain or lose mass? So on your scale, did it gain or lose? You know, what did happen when you did the scale? Okay. What's your reasoning? Obviously, it should be your scale, right? Your scale did this and that, but you know, why did it do that? Is your cell hyper, hypo, or iso? Should be pretty simple. Now, what evidence do you have? Why is it hyper, hypo, or isotonic? Okay, which should go back to number one. Number one should be the reasoning. If a gain or loss, then it's likely going to be, you know, you're using number one to answer number four. And 5, interpret. Now, this is where you have to figure this out in your body. Describe a situation in your body that cup number 1 mimics, and explain why you think cup number 1 mimics this solution. Okay? So these are the questions you have to answer for each cup. Really simple, because you should just already be doing this from your data table and from your models. It's just kind of explain to me what happened. Okay? So it's not super hard to do this, but I want to make sure you understand what's happening. Okay? So you've pretty much got 5 vocabulary terms is what you guys have. You're looking for, you're telling me what diffusion and osmosis is by doing all of this. And then you're looking at 3 of those terms, hyper, hypo, and isotonic. Okay? Those are key to your body. And so you have to explain that to me by doing the model and then with the questions, and that's it. So I know you got it. Okay. I will let you go unless you have questions. So who has a question? If not, you guys are good to go. I want this done by next week because I want to have this all done before Christmas break, so we're ready to go to the next lab. Okay? All right. So if you have a question, stay after. If not, happy diffusing.
All right, bye.
Adios.